information sequencing machines is the .bcl format. These files are binary files containing the base calls per cycle, as well as the quality associated with each call. The advantage of .bcl file format is that each base is recorded as the machine makes the call, which is the most efficient for machine operation. However, if multiple samples were run on the sequencer, then the .bcl raw data must be sorted to separate reads in a process called demultiplexing. This demultiplexing process converts .bcl data into universally used.
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybrid signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read.
Two sets of genes, one from each parent, brought together to produce a new set of instructions and reshuffled in each generation. But sometimes these instructions carry errors, and these can create problems. Faulty genes can cause serious illnesses. Humanity has a long history of figuring out ways to modify genes. We have been crossbreeding plants to make them better to eat for thousands of years. But with gene editing, we've made a great leap forward. This increasingly accurate new technology is faster to use, relatively simple, and cheap compared with previous methods, and promises huge benefits. In a nutshell, it works by identifying then cutting pieces of DNA. One way of doing this uses a component known as CRISPR to pinpoint the precise DNA sequence within the gene to be altered.
Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you can turn a disease-causing all via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene. The same principle applies to gene silencing. A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell. This could be useful for things like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus.
Hi everyone, I'm Kirti Keshav from SRM Institute of Science and Technology. Currently I'm in my third year doing undergraduate biotechnology. Today we had this course on next generation sequencing and I came with almost zero knowledge. By the end of the workshop I kind of learned the basics about what to do in NGS and I'm pretty sure it's going to help me in the future.